All right, ladies and gentlemen, welcome back to the second hub of the Startup Innovation Forum's pitching session. I hope everyone is well refreshed and ready for the second session. We'll be inviting the last 5 presenters of our startups to pitch their innovative ideas to our panel of judges. The top 3 enterprises emerging from today's pitching session will be supported in the form of 28,000 and 100,000 Singapore dollars worth of NAMIC project funding, and the winner will be walking away with $10,000 worth of cash prize, up to $280,000 worth of grant support from NAMIC, and be fast-tracked into the grand finals of Slingshot 2022 as one of the top 50 finalists. Presenters, you are reminded to finish your pitch within 5 minutes and is entitled to 5 minutes of Q&A session each. A bell will be rung at 4 minutes to remind you they have 1 minute left to wrap up. All right. With all the due announcements made, if everyone is ready, let's not wait any further and invite our first presenter of the second session. Now, first in the lineup, let's welcome Matthew Van Heel, founder of Wasna, a biotech startup which formulates animal-free growth serum to provide a cost-effective solution to cell culture process. Matthew, the stage is yours.
Hi, I'm Matthew from Wasna. We are making cell cultures and cultivated meat more affordable. Cultivated meat is an incredible technology. It makes it possible. Oh, it's not working. It makes it possible to grow real meat with such a small ecological footprint that we can do it right in city centers like here in Singapore. Incredible technology. Sadly, it's very expensive, and this is a problem that needs to be solved. Why is it so expensive? It's because you require an incredibly expensive growth medium in order to grow cells outside of a living animal. A growth medium is a mixture of nutrients, the building blocks that cells require in order to grow, and growth factors, which are proteins that tell the cells to grow and multiply. Conventionally, these growth factors, they're derived from animal blood, more specifically fetal bovine serum. Now, of course, it's crazy to make a replacement for real meat when you're still relying on real cows. And that's why every single cultivated meat company has made its main quest to first remove this from their production process. Now, at Wasna, we are helping them achieve those goals. We have developed a patent-pending cell-based technology that produces a serum that is safe, ethical, affordable, and most importantly, it works just as well as fetal bovine serum. Now, our process is quite straightforward. It's a bit like brewing beer with yeast, but instead we use sheep cells to produce a wide variety of different growth factors that you can then use to grow, for example, cultivated meat with. Now, why are people still using animal blood? That is because it's the gold standard in cell culture. If you don't know what your cells require in order to grow, you will give them this blood. Sadly, it's not only unethical, but life science companies, they sell it for a very expensive price point. The bottle you see here is worth $1,200 for a single 1 liter. Now, conventionally, these companies, they try to replace it with serum-free alternatives. Most importantly, they make artificial growth factors using genetically modified microorganisms. These growth factors are usually unique to animal physiology, and you need to combine a lot of different ones in order to create a highly specific growth medium where you can only grow one cell type with. Now, others, they are exploring plant-based solutions. Sadly, plants are not animals, and there are some growth factors that are not in there that are unique to animal physiology. So we have developed a unique technology that replicates natural processes. It works universal, just like animal blood, and therefore we can grow a wide range of different cells with it. We expect to eventually reach a projected price point where we can make cultivated meat at cost parity as conventional meat. Now, due to that, we will help democratize the cultivated meat industry. We will eventually license our technology to large cell culture companies, and we already have 4 different cultivated meat companies that are exploring with our serum. Now, what is very important is, of course, the universality of our product. It's not because you can grow, let's say, skin cells that you can therefore grow muscle cells or something else. And animal serum contains a wide range of different ones, and so does our product. We've now proven to be able to grow cells used to create muscle, fat, milk, offal, and skin. This sparked the attention of a large German life science company that gave us our first contract. They are not making cultivated meat. They are actually using it for wound healing and tissue engineering applications where our technology is also applicable. So next to this incredibly big future market, we expect to be able to get the growth factors from our serum, sell these separately, but right now we can just sell the serum as is for companies that use cell culture, and such as human and animal cell culture. So this is us. Although this project and IP started with my life savings, we are rapidly expanding the team with process engineers and cell culture experts already identified. We also have a wide range of different industry experts across industries. We are looking for roughly $700,000 to get us to the next stage, which is mainly scaling up our production and eventually get the regulatory approval needed for cultivated meat and or pharmaceutical applications. Yes, thank you for listening, and open for questions.
I got one. That was a great presentation, by the way. Really, really good. Who else is doing this, right? And what patents do you have? What IP protection do you have? Because that's...
So it's a European patent application. This is the method of production for the cells and also for actually scaling it up. And and there's also some trade secrets related to how what do you feed the cells in the first place.
And are you already at 85? I assume that's euro cents.
Oh, no, not right now.
That's your target.
It is the target that should be possible within the next 3 years.
And where are you at the moment?
Right now, okay, so the serum itself, you only use usually 1% for cultivated meat production. So $1,200 is for fetal bovine serum. We are now at roughly 253 already. And in order to get this 1%, this 85 cents, you need roughly $60.
Okay, definitely want to talk to you after this.
Okay.
Actually, amazing technology, and I don't want to ask this kind of question already, but how about the regulatory hurdle? Regulatory. So regulation is, of course, always a problem for this kind of, especially this kind of wide range of the application. And you may need to go through one by one, segment by segment.
Yes. So there's quite a lot of regulatory approvals needed. Cultivated meat has different ones from the pharmaceutical industry. We're actually going to work with a large CDMO that already knows how to do these things. They're the only company in the world that actually is capable of selling cultivated meat for human consumption. So they are going to help us with this.
In a sense, as a priority, which country and then which segment can be the most important?
For cultivated meat, this would be Singapore, because it's the only country that currently accepts it. And for pharmaceuticals, it depends. Many of the rules are the same, but likely FDA US is the standard one to go for. Takes also the longest.
Thanks so much. Just a quick question on scalability, without going into the technical aspects of it, but are there any finite resources that you're using where you feel that, bringing this at a very large scale would be problematic, or do you feel there's no particular hindrance there?
So the only reason that it's possible to use a cell-based solution is if you can use standard equipment that is already capable to go to a certain scale. So the largest bioreactors that we can use and that are currently in process, that are currently used for other processes, are roughly 20,000 liter scale. Also in Singapore, there are a couple of those. And we will eventually be able to do this too. Of course, not right now, we're at a far smaller scale, but a 20,000 liter bioreactor should give us a production of roughly 50,000 liter of serum per year, per month. And the total yearly production of FPS right now, every year, is about 800,000. So we would be capable of actually matching this with a single, of course, giant bioreactor.
Sorry, I hope I didn't miss this, but where did the research come from?
This is filed, this was an idea I came up with, and I self-financed this to prove that it worked before looking for outside investment.
Thank you.
So what is the level? What kind of evidence do you have that it's actually working? It's based on just microscopic examination per the slide that there are fibroblasts or there aren't. or are you able to grow cells across a whole scaffold with your reagent?
So conventionally, how you would test these serum alternatives is just using a lab scale, because in the end, the only difference is the scale that you produce at. So for these, we tested on multiple different cell lines. So we tested on fibroblasts, like you say, primary fibroblasts, donor-derived mesenchymal stem cells, intestinal cells, insect cells, and also myoblasts. So,
And how does it compare with traditional media?
Compared to, we make the comparison with FBS because this is the gold standard. We have iterations that outperform it, actually. So it really depends on the concentration that you use. So the more concentrated, the better it will work, of course. But then again, your product will be more expensive. So it's kind of a
How
trade-off.
much are you able to make right now?
Right now, so
Is it 200?
this is the bottle you see here is 200 milliliters. This is a lab scale. So this is very small. This will give you roughly, if you would be making cultivated meat, roughly 20 liters of growth medium. And but then again, this is definitely enough for this big company to do quite a lot of experiments with.
Thanks.
And that will be the end of the Q&A session for Wasna. Thank you, Matthew, and thank you, judges. All right, and next up, we'll hear from Extrabold, an enterprise that specializes in sustainable manufacturing using ultra-large machines with the capability to utilize recycled materials. And speaking to us, let us welcome Yasu Sawada, Chief Marketing Officer of Extrabold. Yasu-san, please.
Thank you. Thank you. Good afternoon, everyone. We are Extrabold, came from Japan. I'm Yasu Sawada, a CMO of the company. We are making super big, ultra-fast 3D printers using PET resin materials. In the beginning, I'm going to talk a little bit about plastic waste. This is the plastic life cycle 4 from OECD's Global Plastic Outlook. In the year 2019, we produced 430 million tons of plastic and 353 million tons turned to be waste. But the rate of recycle is just lower than 10%, and almost 1/3 of plastic waste was generated in Asia. So, and in the case of Japan, more than half of industrial plastic waste comes from industrial sector. This is our focus. But the existing system cannot be the fundamental solution because plastic waste are collected to be mixed and processed by waste collection services. Those services have no incentive in sorting the material, so this leads to material contamination. So, why 3D printers? Challenges in plastic recycling or downcycling and limited application because of these reasons. Familiar 3D printers cannot be the solution. Well, what if PET-type polymer 3D printers? 3D printers, these value propositions listed like flexible supply chain, mass customization, blah, 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 and distributed circular system can turn over the situation to enable upcycling and more applications. Our product, EXF12, can be a solution. There are some features here. Our build area is as big as 1.7 meter, 1.3 meter, and 1 meter. The max throughput is 15 kg per hour. These are some use cases what I can share with you guys. Some chairs, automotive parts, and esports cockpit. But everything's big. Okay, big 3D printers. There are many big 3D printers. It's commercially available in the world. But if you say super big, which has all of X, Y, Z axis exceed 1 meter, there are only 15 models worldwide. If you try to find ultra-fast model, which are pellet resin material, only 4 brands, including us. Furthermore, we are developing next generation printing head based on our proprietary technology, which is mountable on industrial robot and machine tools. Those tools are widely in operation worldwide, as showing annual sales numbers here. We can use this as a channels for promotion, and it will facilitate users' access to the new technologies. Plus, users can set up local distribution, distributed circular system to locally manage recycled plastics without contamination and reduce post-industrial plastic waste. Here is some company snapshot. Our management team is led by Yuji Hara. He's there. He is founder and CEO. He has a deep expertise in machining and additive manufacturing. Also, he has the wide networks in the industry. Some history. In the year 2017, we launched a company. And in year 2020, we won some pitch contest awards in Japan. In last year, we completed Series A funding for 360 million Japanese yen. And later that year, we started commercial shipment of our product. In this year, we showcased for the first time at Japan's largest additive manufacturing trade show. And sometimes in next year, we're going to launch new system with new printing head. Some numbers here. Revenue forecast is 400K in this year ending October, and then 2.6 million next year. We already saw 2 big returns and have 4 backlogs, and we registered 1 patent. This is what we are looking for. US $2 million fundraising to launch our printing head business by expanding our ASEAN footprint through the Singapore organization and increasing R&D resource for print head software development acceleration. That's my story. Thank you.
Thank you, Yasu-san. Over to the judges.
Thank you very much for the presentation. Could you explain again the link between the plastic recycling and the large side printer? There's something I missed in the link, if you don't mind. Thank you.
Okay. As I explained, there's some reason for the challenges in plastic recycling. It's economic aspect and the quality aspect, right? For economic aspect, you guys need to pay more for collection and sorting. So that's why this is a part of economic point. And also quality-wise, in the beginning, I told you guys that one of the problems is the material contamination. But additive manufacturing may have give you guys some value, added value, mass customization, and on-demand production, and reduce the time to market. This can compensate the economic challenge. At the same time, distributed circular systems give the chance to prevent material contamination. Because every local factory manufacturer know well about the history of the material. So that's why you can prevent that. That's the point.
I want to ask you how. Hello, I'm here.
Oh, sorry.
So it's for the recycling process, right?
Yeah.
So do you still need a form of a pallet to use a recycled material, or even you can even use the support structure or failed part to crush it, and then can you put it in as a feedstock? Is it possible?
Possible. We are not only proposing big 3D printers, but also through the partnership with my partner, we are introducing a very compact type shredder and a pelletizer, which every manufacturer can reuse some material to be which is supposed to be thrown. And sometimes you may have some failure in printing, so you can crush, then you can shred without the shredder, and they go back, coming back to the plate to be used at the 3D printer. That's a distributed circular system, right? We are proposing.
Any other questions for the panel?
Maybe one more question, sorry. So I know some players actually have a like exchangeable printer head for both pellet and filament. So it sounds like there might be pros and cons for 2 different types, right? So I'm wondering what is the sweet spot of using pellet-based printer, and then what is the most attractive market applications, and can you give me some example?
If we make the printing heads for filament, there are many competitors in the market, right? And also for the filament, there are more costs for the additional process from the pellet, and also because of this because of scale, smaller scale, then you must pay for a lot of cost for the filament material. But pellet, you can have a more choice at a lower cost. That's the main key reason why we focus on that area. Then we don't think about anything about a filament.
All right, thank you. Any other questions from the panel of judges? All right, if that is all, thank you, Yasu-san.
Thank you.
All right, next up, let us invite Pang Yufei, Chief Technology Officer of Dietrics, a food ingredient innovation company focused on developing nano-functional food ingredients that creates a healthier lifestyle for consumers while ensuring food tastes good. Yufei, take it away.
Thank you. I'm Yufei from Dietrics. So our vision is to create a healthier world where we get to enjoy the food we love but without any worries. So we are excited to explore the possibilities of nano ingredients to create a tasty, healthier food with a sustainable approach. So the problem that we have identified is that obesity is the world's leading risk factor for chronic diseases and premature deaths. So as you can see with these demographics, more than 40% of these Southeast Asian countries are either obese or overweight. So the main reasons is because it is tough to change a habit or give up what they love. Secondly, they're unwilling to compromise on the taste. And lastly, it is due to the socioeconomic situations. So our solution is the cellulose nanofiber, also known as the CNF. It exists in a white crystalline powder form, and it is a joint discovery between Harvard and NTU. So our value proposition is that it does not alter sensory experience. It reduces fat absorption by 36%, and it comes from natural and sustainable sources. So some of the examples that we can integrate our CNF into can be classified into cold, low, and high heat application. The examples are shown on the screen. These market actually combines to form an estimated potential market size of $460 billion US, and these are some of the corporate partners that we can work with. So how it works is that the white crystals will coagulate fat molecules, hence reducing surface area to volume ratio for lipase activity. The CNF is also a stable molecule, hence it will not react with bile salts, and the fats will remain as a whole molecule itself. Lastly, CNF is made up of beta-1,4 glycosidic bonds, and which humans do not possess the enzymes to break down. So comparing to our competitions, oleogel has some problems with integration because of the taste and texture, and inulin actually produces satiety hormones. However, the effect is less direct and usually takes a while before the effect kicks in. So our business model is a direct sales of our CNF to the food manufacturers. Next, we can also create and sell our own end consumer product, or we can also license our IP to other companies to convert their cellulose into our CNF. So this is our ask. We are raising a seed funding of 550,000 Sing dollars. The fund usage is shown on the screen. This year, we are working with a consultant to get a FDA regulatory approval. We are also doing research and development to integrate our CNF into different food products, and we are also looking to scale up with a pilot plant. Next year, we aim to get our first sales and launch our ice cream in Thailand and Singapore. The choice of Thailand is actually strategic because they follow the same FDA regulatory standards. From 2024 to 2026, we aim to have different additives that can achieve different healthier outcomes, and also sell our current CNF into major markets such as Japan, China, and US. So this is our team. Our technical advisors are Prof. Philip, is from Harvard University, while Prof. Ng and Prof. Joachim, they are both from NTU. In terms of business advisors, we have Roger and Clarence with us, myself, the Chief Technology Officer, and Yan Ting is our CEO. So these are some of the media coverage that that's been published about us. And we hope that we can commercialize this technology and make it accessible to the public. Thank you.
Thank you, Yufei.
It's pretty cool, impressive. I'm curious, just from a semantic standpoint, how do you call the food afterwards? Like, you have cell meat-based things, or there is always a branding behind it. Sometimes it's forced by the industry, or sometimes the regulations makes it hard. But you are modifying, obviously, the food, and so do you give it a different name after that?
We can actually label it as low-fat, because what it does, it reduces the fat absorption and you get passed out instead of being absorbed into our body. So there's no special label, but it can be classified as a low-fat product.
There's obviously cellulose is famous for a part of diet sort of ingredients. And do you have any sort of the side effect like feeling bad and something happen, right? If we eat too much cellulose, and then do you have any prevention of such kind of the side effect?
So, if I'm not wrong, you are referring to the Olestra case study, and
exactly.
And we understand that because based on the addition of Olestra, it's quite high. It's about 40 gram per serving. However, for our CNF, we are only looking to add about 1 to 2%. And it can achieve about 30 to 50% reduction in fat absorption. So in terms of the dosage, we are not expecting any side effects. And also we have done toxicity studies in vitro and in vivo, and they are proved to not be toxic.
Thank you.
Is this applied before, during, or after it's cooked, the food?
So this is added as an ingredient. So because it is made from cellulose, so just imagine it as like a powder form, and it can be added into into their dry mix, or it can be added into the cooking or baking process.
And the addition of, let's say, sauces or other condiments don't dilute or block the nanoparticles from attaching to the fat?
No, because in terms of dosage, we are looking at the percentage addition of the final food product. So in terms of dosage, we have proved that it works, and the addition of other sauces will not complicate the matrix. Yeah, so the fat molecules will still be able to bind to the crystal structure.
I'm sure a lot of the big food companies will be very excited about such a product, right? So have you spoken to them, and what kind of feedback are you getting from them?
So actually, we have partnered with an ice cream company. So because of dairy, high fat content, and they are looking to reduce the fat content of their final product. However, we have also noticed. Okay, we didn't notice there was no change in terms of taste and texture. And in terms of their operations, we do not need to change any of their formulation. Just a simple addition is sufficient.
And it's HSA approved?
Currently, we are still pending, we are still trying to apply for the FDA approval. However, cellulose has been used as an ice cream stabilizer since the 1970s. And in terms of particle size, our particles are slightly bigger than what the human body can absorb. So
you don't, with cellulose, you don't need additional approval.
We are getting the approval for the size.
Just a quick question on the choice of your business model. What prompted you to go B2B and not consumer? If it's such a transformative kind of technology, you could choose any of these products and go direct to consumer and capture more margin. Can you share on that? Is it capital constraint?
Yes. I mean, as a startup, we are limited by capital constraint, and we choose to go for a B2B approach to reduce our capex.
In terms of HSA approval and FDA approval, so it's a separate process that you have to go through, right?
So for Singapore, and just now as I mentioned for Thailand, they usually use the FDA as a guide. And once we are okay with the FDA, usually SFA will follow the procedure.
Okay, if that is all, thank you, Yufei, for answering the questions.
you.
And thank you, judges, for the questions. All right, for the next presenter on our list, we would like to welcome Robert Fu, CEO of Cellwave Technologies, a biotech startup which innovates next-generation cell sorter that aims to increase the viability of cells in the sorting process. Handing over the time to you, Robert.
Good afternoon, everybody. The vision we have is to provide the best tools for sample preparation for downstream processing. Our first focus market is actually the next generation sequencing market. As most of you know, RNA sequencing, single-cell RNA sequencing, is gaining a lot of traction in understanding cancer and other difficult diseases down to the genetic level to see what is happening wrong in the cells. However, for it to grow out of the research labs into more widespread clinical care, we see that there are 4 factors. Other companies are addressing items 2 and 3 with a lot of application of AI to interpret all the data. There are various bodies in the US and Europe that is addressing the redemption, insurance reimbursement issues. What we are addressing is actually the tissue sample quality. This slide shows you that the industry standard is actually to have 80% or more of the cells to be viable. If you see the slides, the picture on the right, live cells give you quality data, enabling very reliable diagnosis, whereas a lot of dead cells lead to poor quality data. So that's why 80% is the standard. The unmet need we are addressing is that the current methods of purifying tissue samples are ineffective for fragile cells. In fact, they cause more cell damage than in the process of trying to purify. This is an example of Miltenyi, which is widely recommended as the best method to remove dead cells. You see under graph B that while it is able to achieve more than 80% or around 80% viability, it loses up to 50 to 60% of the cells. Other methods like fluorescence activated cell sorting is also very harmful to the cells because of the very harsh technology. Whereas with Cellwave, with our acoustic microfluidic technology, you see that even with cardiomyocytes, iPSC-derived cardiomyocytes in the right extreme column, whereas conventional FACS dropped 35 to 50%. We actually did this exercise with Beckman-Coulter Mo-Flow machine. Our machine, our device, only loses 3 to 4% viability, and we are able to retain more than 90% of the cells, whereas the methods I told you lose about 50%. And that's a very important thing for clinicians especially, because it means that they only need half or even less of the cells from the patient, especially if the cells are fragile. So this is how it works. On the left is our patented technology, where with a specially unique sound wave, we are able to push the target cells out into the outlet once they are detected by fluorescence. And on the right, you see the high-speed camera recording, where you see every now and then a blip going into the sorted target cells. And comparing with 2 other recently launched cell sorters, which also claim high viability, what we have is a product that is able to address a whole range of human cells and at a speed that is very suitable for single-cell sequencing. The Wolf product, as you see, is too slow at only 300 cells per second, and the Taito product is limited by the size of the cells that they can sort. So as a go-to-market, we want to position ourselves as a first step in the workflow in single-cell RNA sequencing, and partnering with products like 10x Genomics and Mission Bio and so on to be the first step in the process. And our next milestones, we are actually right now tidying up the POC prototype for field testing by the end of this year. And with successful field testing, we want to raise second round, Series A, for design for manufacturing, and at the same time fund some early work on version 2. Based on market data from 10x Genomics and others, we estimate that if we are able to reach 40% of the 10x Genomics installed base by the 5th year, our revenue will be that number that you see. And note that more than 50% of the total revenue is recurring revenue. So our ask is actually for $1 million seed funding, of which we have closed 400,000. And I hope you can support us in getting to that stage. Thank you.
Thank you, Robert. Now we're open to questions from the judges.
What is your distribution strategy? Because if this is as amazing as your competitive analysis shows that it is, then the next challenge for you is how do you get this into market? So what is your thinking around that? And then how does your pricing compare with the other technologies, and do the buyers really care about the sorting rates that you do versus their inferior rates?
Well, our beachhead market is very important. We want to focus on researchers who are working with fragile cells, which are small quantity. So liver cancer cells and cardiomyocytes and so on. So if we can conquer that beachhead, then we can then spread the use into other cell sorting needs, because it's the same technology that all the other cell sorters will be competing for. But that's too big a competition out there for a small company like us, just bringing one module of the whole workflow. So I think the preferred strategy is really to partner with the people like Bio-Rad, Mission Bio, whom we have talked to sub-license or even to customize a version that will fit seamlessly into their process.
Can you come back to the team slide perhaps and tell us your story, because it sounds like your team has a lot of experience about that field.
Which one?
The team slide?
Oh, the team, yes. Yeah. Okay. Professor Ye Ai is the inventor and the founder. He's been working on this acoustic microfluidic technology for more than 10 years with MIT, Los Alamos, and so on. Los Alamos. Lawrence Lim is our engineering advisor. He's got a lot of experience in producing very precise microfluidic engineering products. He came from Becton Dickinson, actually. And Dr. Krishan is our research advisor. He's been working on iPSC-derived cardiomyocytes and identified the need for something like our product, because they are not able to extract those products, those cardiomyocytes with conventional cell sorters. And I have a lot of business development experience and was introduced to this team, and we formed the company 3 years ago.
So this is, it's under patent prosecution. I mean, you filed for patent protection.
It's been granted in the US and
granted already.
And China.
Okay.
Yeah.
And how much patent life is left?
Well, we have a exclusive license from SUDD for the life of the patent.
Right. What's the balance of the life of the patent?
The life of the patent.
Not sure.
Question I have not explored. Maybe it's 25 years.
Yeah.
2020.
All right. So quite a long road. Good. This
yes.
Good balance.
Any further questions from the judges? All right, if there are no further questions.
Thank you.
Thank you, Robert.
Thank you.
All right, very quickly now we're down to our last startup for today. And for our last startup for today, let us welcome Reacto, an enterprise that aims to provide a solution to treating industrial wastewater by utilizing an electrochemical process that's cost-effective and sustainable. Pitching their ideas today is co-founder Massimo Spina. Massimo, you may begin.
Thanks. So, good afternoon, everyone. I'm Dr. Massimo from Reacto. And together with my co-founder, we combine 20 years of expertise in material science and wastewater treatment to create a patented breakthrough solution to recycle the hardest to treat industrial wastewater that is currently incinerated. This is industrial wastewater. It's highly toxic both for humans and the environment. The toughest to treat contain persistent organic pollutants, highly toxic and non-biodegradable substances that are present everywhere. Because of their bioaccumulation, worldwide, rainwater is no more safe to drink. And it gets even worse. Even unborn babies have these forever chemicals in their bloodstream before they are even born. This is why, globally, companies are getting heavily punished and even face jail terms for non-compliant discharge. But why is that? Why would they risk that? Standard treatment for now is to concentrate this wastewater on site, then truck it off-site for incineration, then send incinerated ashes to the landfill. Yes, people are literally burning water, very energy-intensive and expensive, because at the moment it's the most cost-effective way to dispose this type of waste. Globally, each year, more than 70 billion tons of wastewater with POPs are produced, and the treatment with alternative to incineration is worth more than $7 billion. Reacto's patented technology combines graphene with electrochemistry to create a solution who, just by using electricity, breaks down the toughest organic pollutants, enabling the recovery of the wastewater. We are the first company in the world to atomically bind graphene on the surface of the electrodes of an electrochemical system to double the energy efficiency to decompose the toughest organic pollutants. Our patented technology, exclusively licensed to Reacto, enable companies to safely treat the waste on site, eliminating the need for transportation, maximizes the water recovery, and doesn't create any sludge or waste that needs to be sent to the landfill, all while saving cost up to 50%. This is an example of our technology at work. This is a wastewater coming from a faster-moving consumer good company, Fortune 500. Concentration of organic pollutants is roughly 100 times more than what you will normally see in a standard wastewater streams. Going through our process, just by using electricity, you can see in the left side, transparent, clean water, safe to discharge, no sludge, no residues. We tested our technologies with more than 10 types of wastewater streams, come from 3 different industry verticals: chemicals, pharmaceutical, and FMCGs. Our technology has other competitors, but electrochemical oxidation process in particular, are proven to be the most cost-effective way compared to incineration, chemicals treatment, or other technologies like photocatalysis and oxidation. Because of our patented electrodes, Reacto is 2 to 3 times more energy efficient than a standard electrochemical oxidation process, and that's our edge. We already finished a paid proof of concept with a Fortune 500 company, and we are now working with 7 multinational companies in Americas and in Asia to test our technology. We are now raising our seed round to prove our technology at scale and validate our business model by next year. We are reaching our market directly or in partners with water system integrators to leverage on their existing network of clients and industrial expertise to speed up our time to market. To monetize this technology, we are offering a treatment as a service, when we will build, own, and operate the system on site in the manufacturing plant, charging the customer based on the savings on the treatment that they are having at this point in time. Alternatively, we're also selling our system and getting recurring revenues from maintenance and replacement of our parts. This is the team, is myself and Dr. Zheng Xiong. We were 2 scientists from the National University of Singapore, and we have the full support of our 2 founding labs, both in terms of expertise and resources. Our technology was not easy to make. It took 7 years of deep tech R&D invested by the National Research Foundation, NUS, and other government entities to be developed. After our incubation, in only 1 year, we went through the Shell Startup Engine, won some awards with the Real Tech Award, and we just came a few months ago as a global clean tech energy champion from the Extreme Tech Challenge at Berkeley. Starting from Singapore, Reacto aims to become the sustainable solution for industrial wastewater that is currently incinerated, so we can save precious water resources, eliminate harmful pollutants from the environment, all while reduce energy consumption and minimizes greenhouse gases emission. We are looking for partners and investors to join our mission. Speak with you soon.
Thank you, Massimo, and over to the judges.
Can you talk a bit more about the power requirements of the machine and how it works generally with energy?
Yes. So energy is 90% of our consumable, or even more, 95, 99%, I would say. It depends on the type of wastewater we are treating. So it depends on the concentration. So normally municipal wastewater have very small load of organic pollutants. So I say even up to 100 times or even more than that in terms of pollutants. So the energy consumption you have normally for water desalination is about a few kilowatt hours. In our case, it can be few kilowatt hours in terms to break down the toxic pollutants, but not completely decomposing it. If you want to do a complete decomposition, it can go up to a few thousand kilowatt hours, but you consider that in the terms of the cost that people are paying to store the waste on site, track it off site, incinerating, disposing the ash, you're still talking about at least 50% cost savings. Another way to look at it is this way. So we put US dollar per kilogram of organic pollutants removed. That's a more cost-effective way to put it, to compare 1 meter cube of wastewater that is municipal or 1 meter cube of wastewater that is industrial, highly concentrated. This is a recent study from 2021 where they compare all these main technologies. Photocatalysis is UV, normally you have for disinfection. Ozonation is very good, but it's highly expensive in capital cost. Incineration is cost-effective once you concentrate the waste. Cheapest so far is chemical. Chemical, you just take hydrogen peroxide, pour it into the water, create a lot of toxic sludge you still need to dispose. Our system, like a normal electrochemical process, apply electricity, break down completely the pollutants, or increase the biodegradability of the pollutants, and you're done.
What will be the maintenance cycle cost for the maintenance?
So the maintenance cost, our electrodes, compared to other electrochemical processes, are self-cleaning, meaning that you're locally generating these highly reactive species who break down the toughest organic pollutants. So our lifetime of the electrode is at least 5 years. So far we make a joke, they never die. So at the beginning when we were testing wastewater, we were using this one clean electrode for every type of waste. Now we just use for every type of waste, and still the performance have not been degrading.
No using almost no consumables, and also no need to replace quite.
No, yes. Yes.
Maybe a bit of a provocative question, but how far do you want to take this company? Because we've invested in water, and the typical pattern we've seen is that water is one of the hardest problems to crack for technology startups because they have slow adoption, high capex, and they end up essentially being bought out fairly early by a bigger player who has the balance sheet to bring it to the industrial clients. So I'm just questioning, it's not so much about the superiority of the technology, it's much more around the economics of it. So hence my question, how far do you want to take this company and to which stage?
I would say we go IPO. Very clear from my company, with my co-founder, we are clear if you want to make it, we want to make it big. What inspires us is that we want to take deep tech and commercialize it. So wastewater and persistent organic pollutants is a huge issue. Now in the US and Europe, everyone is talking about PFAS, is a big topic about that. So we are going to use this as a launchpad for our technology. But what you're having also is a deep expertise in graphene and material processing. That is something that would also eventually be spun out in other applications later on for our expansion phase.
Yeah, sorry again, just to rephrase. Issue in water is no longer about the superiority of technology or the need. It's around the economics of being able to sustain a company with investors to a price point and a scale by which it's economically viable. So my question is, again, so I was asking maybe a little bit provocatively, how have you thought about this problem? How do you bring that company to IPO through the different investments that's needed? Just, it's a long question.
Yeah, so it's a capital-intensive process. That's why we're offering a treatment as a service to engage company earlier, because they don't need to have this initial capital cost if they want to buy in. So we're going to own the system and just maintaining and charge on a treatment as a service based. And that.
Sorry to interrupt, but as a service makes it even more costly. You have to acquire the asset, you have to get financing for it. It's even more costly to offer it as a service than as a standard product. Just not very clear.
Yes, it's going to be initially, but we're not going to start with a huge project at the beginning. So we're going to have the cash flow in. Our payback period is less than 2 years' time. So we are going to make sure that the capital cost is recovered quite fast. At the same time, the fundraising for the Series A and so on will be enough to finance our first project.
And thank you.
How does this compare with algae-based treatments, things like that? Because the benefit with an algae-based approach, we have a company that's essentially doing wastewater treatment using algae, but from that they're also generating carbon credits, right, and feedstock. So how does this compare with that kind of approach?
What you're kind of focusing is these persistent pollutants that are not biodegradable. That's why they call it forever chemicals, because the lifetime is thousands of years. That's why newborn babies get it, rainwater gets it.
So is there anything that this doesn't filter?
It's not a filter. We just break them down. So there is no filter that gets clogged.
Okay,
it's
let me rephrase. Is there anything that gets through this?
No. So we just kill them as long as we want. So our goal is to break them down, to make them non-biodegradable, so any process can easily treat it.
So just water molecules coming out the other side. There's no minerals, there's no anything else.
Oh, the inorganic part is being filtered out, will be filtered out. Okay. What you're focusing on is persistent organic pollutants.
Okay.
That are the nasty beast in the wastewater business.
Okay, thanks.
All right.
So just to follow up on Phil's question.
Yes.
These persistent organic pollutants, are they entirely cleared? I mean, how long does it take, let's say in 100 mils of water?
We are talking about, we can break down the pollutants in few hours. If you
bring
want to
down by what, 70%, 80, 90? Or?
Okay, so we can break down the chemical chains that are difficult to attack by biological bugs. That can just take few hours. Okay, if you want to completely eliminate the pollutants, that might take maybe 1 day.
Mm-hmm. And it works across all
organic
pops.
Pollutants, all pops.
Thanks.
All right, thank you, judges, for the question, and thank you, Massimo.
Thank you.
All right, ladies and gentlemen, that concludes the startup pitching session, and we definitely have heard some very exciting ideas that will prepare us for a more sustainable future. We'll now adjourn and allow the panel of judges time for deliberation. Meanwhile, I'd like to invite all guests and startups to take a short break and be back by 5:45 for the results and prize presentation ceremony. I'll see all of you then. Thank you.